Hi students, today we can discuss about the metabolism of proteins. Proteins are large macromolecules with one or more mixed polymers of amino acids called polypeptides. Proteins consist of carbon, hydrogen, nitrogen, oxygen and sulfur. In addition to these, some minerals like phosphorus and iron are also present. Protein constitute 50% of dry mass of protoplasm. Let us see what are the sources of proteins. Proteins are or the major sources of proteins include fish, egg, lean or fat-free meat, milk, curd, cheese and beans or leguminous plant products consist of more amount of proteins. Maximum amount of protein is present in soya bean. About 43 to 45% protein is present. Milk consists of only 4% of protein while egg consists of 13% of protein. Proteins are made up of 20 amino acids. As you know, they come under two categories, essential amino acids and non-essential amino acids. Non-essential amino acids are 12 in number and essential amino acids are 8 in number. Human beings also synthesize 2 amino acids at a slower rate than their requirement in the body. They are called semi-indispensable amino acids. Example, arginine and histidine. Metabolism of protein means the constructive and destructive process of protein. Protein undergo metabolism in two ways. One is called catabolism, means breakdown of protein molecule, that is otherwise called destructive process which release energy. And the other one is anabolism or the constructive process which utilize energy and produce larger molecules for cytoplasmic membrane, cell wall, ribosomes, etc. When we examine the general pathway of amino acid metabolism, see, amino acids are formed as a result of digestion of proteinaceous food and as well as metabolites of glycolysis and Krebs cycles. So amino acids will undergo two pathways. One is the anabolic pathway, means synthesis of cell wall extracellular protein, then synthesis of peptide physiologically active substances and form protein and peptides of the organism. The catabolic pathway include transamination, deamination, decarboxylation, etc. to break down the amino acids in finally to carbon dioxide, water and urea. The first step is deamination. Elimination of amino group from amino acid with the ammonia formation. There are four types of deamination process. One is oxidative deamination, reductive deamination, hydrolytic deamination and intramolecular deamination. Oxidative deamination takes place in presence of L-glutamate dehydrogenase, which present in cytosol and mitochondria of liver, and L-glutamate is also take part in amino acid deamination. In reductive deamination, amino acid undergo a convert into a fatty acid. Like in hydrolytic deamination, amino acid will convert into hydroxy acid, and in intramolecular deamination, amino acid will become unsaturated fatty acid. The next step is transamination of amino acid. The process of transferring of an amino group from an alpha amino acid to an alpha keto acid is called transamination. Here actually transamination means conversion of amino group from one amino acid to another. The enzyme that involved is aminotransferase or otherwise called transaminase. There are different reaction of transaminases between alanine aminotransferase and aspartate aminotransferase. See here, amino acid convert into 2-oxoglutarate, alpha ketoglutarate into 2-oxoglutarate. Aminotransferase pyridoxal phosphate is present, or the 2-oxo acid will convert into then to glutamate. So this is a reversible reaction. Here we can see how the transamination occurs in presence of amino transferase. For example, alpha amino acid and one alpha amino acid combined with alpha keto acid and form another alpha keto acid and another alpha amino acid. Example, alanine plus alpha keto glutarate. In presence of alanine aminotransferase gives pyruvate and glutamate. Like another in presence of aspartate aminotransferase, here aspartate plus alpha ketoglutarate will give oxaloacetate and glutamate. These two are the examples for amino, that is transamination process. The next one is decarboxylation of amino acid. Means here the removal of carboxyl group from amino acid into carbon dioxide. So that is why the process to remove of carbon dioxide from the amino acid with formation of amines. So the product is the amine. The enzyme that involved is decarboxylase. The coenzyme is pyridoxal phosphate. So here what will happen? One amino acid will convert into amine, corresponding amine by releasing carbon dioxide or removal of the CO2 from COOH group, and which will become the amine. So this is called a decarboxylation reaction of amino acid. Dear students, from this class I hope you could understand the metabolism of protein. See you later in another class with another topic. Till then, bye. Thank you.